Okay, I invite you today to this lecture, and in this we look at how a certain group of living things regulates or decides which genes to express and which genes not to express. This group is made up of those organisms that we refer to as prokaryotes. I'm doing this video from a hotel room. As you know, I'm undertaking some field sampling, so don't mind my background. So we've seen how we as eukaryotes regulate which genes to express and how we express our genes, how we create proteins. We saw that in our last lecture. But this process of gene expression within prokaryotes is fairly regulated in a very tight way. And why that is important is the tight regulation helps to increase the efficiency with which the resources that are found within the cells of these particular prokaryotes are used. Most of the way these resources are used with respect to prokaryotes is with how they respond to the changes in their immediate environment. So how the expression of these genes is regulated in prokaryotes, it allows them to adapt to the different conditions within their immediate environments, and with that it helps them to produce proteins only when those proteins are needed. Okay. For us as eukaryotes, we'll have an entire topic called epigenetics. Okay, and we'll see how that happens. So the mechanism of regulating this gene expression within prokaryotes includes a regulation of the transcriptional mechanism. So you remember that transcription refers to the first step in the gene expression mechanism, and it is this step that leads to the formation of an RNA transcript. So transcription leads to the formation of an RNA molecule using a DNA template. So in prokaryotes, the regulation of this transcription is the most common form of gene regulation. So which means for the prokaryote to regulate the expression of its genes, it just regulates the transcription mechanism. Okay. Once it deals with the transcription, it has dealt with how the genes will be expressed. So among the key features within the transcription regulation among or within or in prokaryotes, they include several things. One, we have a feature that we call a promoter. Okay. So remember, we are trying to regulate transcription, which means we are using DNA to create RNA. So a promoter is a sequence within the DNA molecule that serves as a binding site for the RNA polymerase. RNA polymerase is what is initiating the process of transcription. Because as an RNA polymerase, it leads to the formation of an RNA polymer. By forming an RNA polymer, we will have done a transcription. Okay? So the site in the DNA where the RNA polymerase will go and bind or attach, that is the promoter. So because these promoters are recognized by the RNA polymerase, then it means these promoters must be having specific sequences onto which the RNA polymerase will go and bind. So the RNA polymerase itakuwa inatafuta that specific sequence, more like the key lock hypothesis. Then another key feature is what we call an operon. So within operons, you have genes that are organized, genes that are grouped together. Okay, so you have genes that are grouped together because they have related functions. So by those genes being grouped together, by virtue of their related functions, then those genes have to be transcribed together. Why they end up being transcribed together is because they have a single promoter unit. So one promoter controls several genes, and those several genes controlled by a common promoter, those common genes have related functions. So when you have several genes grouped together and they are being controlled by a single promoter, then they are known as an operon. Okay? So it means an operon then will be made up of the promoter, which is controlling all those genes. There will be an operator. An operator is a section where some regulatory proteins will be binding. Then in addition to the promoter, the operator, now comes the genes, which are grouped together. Okay, so an operon has the promoter section, it has an operator, then it has the genes which are controlled by the single promoter. Still within the transcriptional mechanism or the transcriptional regulation within prokaryotes is another feature that we call a repressor. To repress is to inhibit. So repressors are proteins which will bind to operators. And once these proteins have bound to the operators, they will inhibit the transcription of the genes that are found within the operon. Okay? So a repressor refers to a particular protein which will bind to an operator. And once that protein has bound to the operator, it will lead to the inactivation, okay, inhibition of the transcription process of those genes that are found within the operon. Remember, within the operon, you have a promoter, you have an operator, and you have the what? The genes that have to be transcribed. So kazi ya repressor inashikana na operator. Promoter kazi yake ni kushikana na RNA polymerase. The other component or the other feature that is vital in the transcriptional regulation of prokaryotes is the activator. An activator does the opposite of a repressor. Okay? While a repressor inhibits transcription, an activator enhances transcription. Okay? So within the prokaryotes, there are some genes that will require an activator protein. Okay? So what the activator does, it once it binds, then it enhances the transcription, and we'll see how that happens. Okay, so once the activator binds, it leads to the efficiency of the RNA polymerase kushikana na promoter. RNA polymerase ikishikana na promoter, hapo ndio itasomwa, transcription itafanyika. Okay, so kazi ya activator ikishaenda ibind kwa operon, section of the operon, inaongeza the efficiency with which the RNA polymerase can actually lead to the reading or the transcription process. We have inducible and repressible systems. Okay, so prokaryotic gene regulation inaweza kuwa classified ama categorized either as being an inducible system. With that, it means that genes are usually off, but they need to be induced. They need to be turned on once an inducer is present. On the other hand, a repressible system means the genes are usually on, but they can be turned off once a repressor molecule is present. Inducible system, the gene is off, but inahitaji something to turn it on. So once an inducer is present, then the genes that are usually off can now be turned on. Why? Because an inducer is present. On the other hand, for the repressible system, in this case, the genes are usually on. Ni kama switch ya stima iko on, but you need to turn it off ndio uokoe tokens. Okay? So repressible, the genes are usually on. Something needs to be present so that they can be turned off. Okay. Now, what is an operon? An operon is a group of related genes mostly found in a bacteria. And these genes are regulated and expressed together. So most of the time you'll find that these genes work together to perform a specific task. Such tasks will include, for example, either breaking down a particular type of sugar or building a particular type of protein. So the system of an operon then will allow the bacteria to control when this particular group of related genes can either be turned on or off, basing on the environment and the needs of the cell. If the environment and the needs of the cell command kwamba these genes should be turned on, maybe because the product of the genes is needed to break down a particular sugar, then those genes will be turned on. All of them will be turned on. What then are the components of an operon? We had seen a few of those. One is an operator. An operator is like functions like a switch region where the repressor protein can bind. On the operator, hapo ndio repressor inakuja inashika. Once the repressor imeenda ikashika kwa operator, then that repressor can either block the RNA polymerase from continuing with the transcription. Okay? So repressor imekuja imeshika operator. Once the repressor binds to the operator, then transcription will not happen because the RNA polymerase haitaweza kusoma the entire DNA region so that it can create an RNA transcript. Then there is a promoter, which is a region where the RNA polymerase, which is the enzyme that is leading to the formation of an RNA transcript. So the region where the RNA polymerase can actually bind so that the process of transcription can begin. It is the process of transcription that is now leading into the formation of an RNA transcript, in this case a messenger RNA. Then it is the messenger RNA that will later be used to produce proteins through the process of translation. Munakumbuka the genetic code. So pahali kwa DNA ambayo RNA polymerase itaenda ishike ama ijiattach, so that after imejiattach ndio ianze kusoma the DNA, leading to the formation of a messenger RNA. That site, that region is what we call the promoter. We've talked about a promoter, we've talked about an operator. So it means the RNA polymerase needs to read some genes. So the actual genes that can code for proteins, those are now the structural genes. Those are the groups of genes that are being controlled by a single promoter. Okay? And then at times, we have a regulatory gene. And this gene, most of the time, is located outside the operon. It's not part of the genes that are found within the operon. But it is that gene now that produces a repressor protein. Once that gene has produced the repressor protein, that repressor protein ndio itakuja ibind kwa operator. Okay? So the repressor protein will control whether the operon is either on or off. How does it do that controlling? It does it by binding to the operator region. If it binds to the operator region, then the RNA polymerase cannot move along the DNA sequence. Lakini hiyo repressor ikikuja itolewe baadaye na kitu tutaona. I don't want to preempt it now. Once the repressor is removed from the operator, then the RNA polymerase can actually now move along and transcribe the DNA into a messenger RNA or the RNA transcript. Then that RNA transcript, sasa hiyo ndio inachukuliwa, inapeleka kwa ribosome. Then inside the ribosome, a protein is formed. It is a protein that has function. For example, the enzymes. Okay. We move on now, and what we see here is a diagrammatic representation of an operon and the entire process that is happening. In this diagram, you have a regulatory protein. Hii ndio inatengeneza a repressor protein, this regulatory gene. This one is located outside the operon. The operon inaanzia hapa kwa promoter ikienda huku. Is from the promoter to the right. But the regulatory protein, the regulatory gene is located outside the operon. So kazi ya hii regulatory protein ni kutengeneza a repressor protein. Then that repressor protein kama imetengenezwa na iende ibind kwa operator. Then it is the promoter region penye the RNA polymerase inatakana kukuja ijiattach. Once the RNA polymerase has attached itself at the promoter region, then that RNA polymerase can now begin the process of moving along to transcribe the genes. But hiyo RNA polymerase ikikuja iattach kwa promoter, lakini repressor imetengenezwa na the regulatory gene, na hiyo repressor imeshika operator. Hiyo repressor operator venye imetengenezeka, imeshikana hapo, itasimamisha the RNA polymerase kusonga so that iende isome the structural genes. Unless the repressor protein itolewe kwa operator, transcription cannot happen. Okay? It is until the repressor protein imetolewa, hapo ndio sasa RNA polymerase itaenda isome those structural genes. Ikitembea hapo, inatengeneza the messenger RNA through the process of transcription. That messenger RNA ndio itapelekwa, itoke kwa nucleus, iende kwa cytoplasm inside the ribosome, na hapo ndio tutakuwa na hiyo kusomwa kwa amino acids. The amino acids will be joined together to form proteins. Remember, these are structural genes which are part of an operon. Being structural genes, they are controlled by one promoter. So all of them are either turned on or all of them are turned off. Once they are turned on, it means they are transcribed. So they form a messenger RNA. The messenger RNA then is translated to form the various proteins. So in this case, gene X will form protein X, gene Y will form protein Y, and gene Z will form protein Z. Okay? So as we indicated, there are two types of operons. One is the inducible operon, and two is the repressible operon. We've seen that within an operon, there are several genes that are controlled as a unit by a common promoter. So then, are operons important? The answer is yes. And one of those main vital points of why operons are important is they help in energy efficiency. So, by virtue of all the genes being either turned on or off, it means then operons will help bacteria to save energy because genes are turned on only when they are needed. So the bacteria cannot produce a protein which is not needed. So kama protein inahitajika, itatengenezeka. Kama haihitajiki, haitatengenezeka. Tutaona hiyo as we proceed. The second importance is that the operons help the bacteria to cope with the environment. They aid the bacteria to adapt to the environment. So there is need that a bacteria adapt to the environment because the environment will keep changing. If the bacteria can't adapt, then it will die. It will not survive. Okay? So on the basis of the needs of the environment that are keeping to change all the time, the operons will allow the bacteria to adjust quickly, either by turning on certain genes or turning them off all at once, largely depending on what is available in the environment. If, for example, a certain sugar is available, then the bacteria can now turn on an operon to create enzymes that will help to break down that particular sugar. The sugar is available in the environment of that bacteria. The bacteria then turns on the operon. Okay? Once the operon is turned on, then the genes within that operon are transcribed and eventually expressed through the formation of proteins. The proteins will most likely be enzymes which will help to break down the sugar that is present in the environment. If the sugar that was present in the environment is no longer present, then the operon or the bacteria will turn off the genes. The genes were turned on because the sugar was present. But now that the sugar is absent, the operon, the bacteria will switch off the genes. So there will be no enzyme formed because there is no longer sugar that is present because the environment has changed. So think of an operon like a factory, and the promoter is the factory gate where the workers can enter to start making products. In this case, the workers can be the RNA polymerase. Then think of the operator as the switch that controls whether the factory is open or closed. Okay? Because remember, the RNA polymerase can only move if the operator is not occupied. If we think of the operator being occupied or having a repressor on it, then by the operator having a repressor, then we can think of it as the factory being closed. So once the factory is closed, the worker will not move in. Ukitaka kwenda library na upate mlango imefungwa, hautaingia wapi? Kwa library. Okay? Then think of the repressor. Ikuwe ni kama ule mtu mwenye anafunga, block the entrance. So once the repressor imeenda imeshikana na operator, then the entrance imefungwa. The worker in this case, which is the RNA polymerase, will not be able to come in. When the RNA polymerase has come in, itaenda isome the genes, then itengeneze protein. But kama the worker, wewe ukienda kuingia kwa university ya Lupe, upate wamefunga gate. Okay? Upate security guard kwa gate amefunga na anakuambia hauingii, hautaweza kufanya kazi, hautaweza kuenda usome, hautaweza kuenda attend lecture. Okay? So using this analogy, then you can easily now understand what the promoter does, what the operator does, and what the repressor does. So in summary, therefore, operons help bacteria to control when to turn on or off a certain group of genes dependent on the needs of that particular bacteria. The operons are made up of promoter, operator, the genes within that are either supposed to be turned on or off, and another regulatory gene that is outside the operator. Kazi ya the regulatory gene ni kutengeneza the repressor protein. Okay, regulatory gene kazi yake ni kutengeneza the repressor protein. The inducible operons are turned on by an inducer. Repressible operons are turned off by a co-repressor. Okay? So this system of operons, this operon system, then helps in the regulation of genes. It makes that regulation to be efficient and to be responsive to the environment. Efficient sababu hakuna wastage. A gene is only being expressed kama kuna haja. That is efficiency. Then responsive because the gene is being expressed in response to the change in the environment. So let's look at these two operon systems. One is the inducible operon. Tumeona kuna an inducible operon, kuna a repressible operon. So an inducible operon is a group of genes that are normally turned off. So normally, this group of genes zinakuwa off. But the group of genes that are normally off can be turned on when something specific is present in the environment. An inducer, ikikuwa present in the environment, then the group of genes which are normally off can now be turned on. An example of an inducible operon is the lac operon. Okay? This lac operon is found largely in the Escherichia coli, which is a bacteria. It's part of the normal flora within our intestines. So it is called a lac operon because it helps this bacteria to break down lactose, which is a sugar that is found in milk. Okay, so that is how the name LAC, L-A-C, comes about. So how does this lac operon work? Because it is an inducible protein, inamaanisha it is normally off. The normal state of this lac operon ni kwamba it is turned off. So when there is no lactose around, there is no need of having the lac operon turned on, sababu hakuna lactose. So this is because there is a repressor protein that will be bound to the operator region of the operon. The presence of that repressor protein, ikienda ishikane na operator region, then the genes will be prevented from being transcribed by the DNA-dependent RNA polymerase. It is a DNA-dependent RNA polymerase, sababu it is an RNA polymerase enzyme that is creating an RNA transcript using a DNA sequence. That's why tunaita DNA-dependent RNA polymerase. It is creating an RNA polymer by depending on a DNA sequence. So it is normally off. But when lactose is present, then the lactose molecule will act as an inducer. That is why it is an inducible operon. So why is the lactose molecule being called an inducer? It is called an inducer because this lactose will bind to the repressor protein. Remember, what makes the lac operon to be off ni kwamba there is a repressor bound to the operator region. But once there is an inducer, which is lactose in this case, hiyo lactose inaenda inashika the repressor protein. Ikishika the repressor protein, yenye imeshika it is already bound to the operator. So the inducer inaenda inashika the repressor which is already bound to the operator. And once the inducer touches or binds to the repressor protein inabadilika in shape. Na ikibadilika kwa hiyo shape, inapoteza the ability to continue attaching itself to the operator. So inatoka. Kitu yenye imesababisha repressor protein ikatoka kwa operator ni lactose yenye ilikuwa present in the environment. Venye ilikuwa kwa environment, ikaenda, ikajiattach kwa repressor, which was already present on the operator. That is why the lac operon was off in the first place. But once the lactose molecule has gone and bound to the repressor protein, which is present on the operator, that repressor protein changed its shape. Once the shape changed, it led to the removal of that repressor from the operator. Once the repressor has left the operator, then now the DNA-dependent RNA polymerase can now move and transcribe the genes within the operon. Once the genes have been transcribed, then we will get an RNA transcript. Then out of that RNA transcript, we'll end up getting proteins through the process of what? Translation. Okay? So with the repressor protein now no longer blocking the operator, the DNA-dependent RNA polymerase can now transcribe the genes in the operon. Once this transcription has been done, then the production of proteins that are responsible in the breakdown of lactose as an energy source will be accomplished. So in this figure, we are showing the lac operon, which is an inducible operon. As we described earlier, in the absence of lactose, the lac repressor binds the operator. Once the repressor is bound to the operator, then the RNA polymerase is blocked from transcribing. But if lactose is present, in this case lactose is an inducer, then the lac repressor is released from the operator. Why? Because lactose inaenda inajiattach kwa hiyo repressor. Once it attaches itself to the repressor, the shape of the repressor changes. Once the shape of the repressor changes, the repressor then inawachilia the operator. Once the repressor has let go of the operator, then now the RNA polymerase can transcribe. The product of that transcription is an messenger RNA. Then that messenger RNA will be translated on the basis of the sequence to create amino acids. Then those amino acids will be joined together to form proteins. So the key points to note here is an inducible protein is always turned off until there is need. What will lead to the turning on is the presence of an inducer. And how an inducer turns the operon on is by inactivating the repressor. Once the repressor has been inactivated, itawachilia the operator. Then once the operator is free, now the RNA polymerase, which will have already attached itself to the promoter, will move, transcribing the genes, creating an RNA sequence. Then that RNA sequence will now be translated into proteins. Those proteins will have a function, okay, because enzymes are protein in nature. So a quick summary: no lactose. If there is no lactose, then the repressor blocks gene expression. If the gene expression is blocked, then the operon is off. In the presence of lactose, that lactose will bind to the repressor. Once lactose has bound to the repressor will fall off the operator. Once the repressor has fallen off the operator, the genes will be expressed. So by expressing the genes, we will have turned on those genes. So through this way, the cell only makes the enzyme to digest lactose when lactose is present. So it means it only makes the enzyme to digest lactose when that enzyme is needed. So by so doing, it saves energy and it increases efficiency. Let's now look at the repressible operon. So a repressible operon, as you will guess, is the direct opposite of an inducible operon. So a repressible operon is actually turned on, is always active, but it can be turned off when a certain molecule, in this case a co-repressor, is present. An example of this repressible operon is the trip operon. tryptophan is a good example, na hiyo inapatikana pia kwa E. coli, na hiyo inachangia kwa kutengeneza this amino acid that we call tryptophan. Okay, so it helps E. coli to make tryptophan, which is an amino acid. So how does the trip or the repressible operon work? So this repressible operon is normally on. So when there is low tryptophan in the environment, then the trip operon is turned on. Okay. So remember, tryptophan iko very low in the environment. So kumaanisha the bacteria needs to turn on so that it can lead to the formation of more amino acid, because it is already low in the environment. Okay. So this means the genes are being expressed and the cell is making tryptophan. When there is low tryptophan, there is need kwamba watengeneze more tryptophan. But now what happens when tryptophan is present? So kama tryptophan haiko in the environment, ama iko lakini at a very low concentration, then there is need to turn on this operon so that we can make more tryptophan. But when tryptophan is already present in an enough concentration, then that tryptophan haitakikani kutengenezwa sababu tayari iko. So the tryptophan in this case will act as a co-repressor. Hiyo ndio itaenda sasa, it binds to the repressor protein. By binding to the repressor protein, it will activate that repressor. Once the repressor is activated, that activated repressor will now bind to the operator. Once the repressor has bound to the operator, it now leads to blocking the operon from being read or transcribed by the RNA polymerase. Once the repressor imeenda ikabind kwa operator, the RNA polymerase cannot read the DNA sequence. So by doing that, then it cannot transcribe it. So it means the gene transcription has stopped. So with the repressor bound to the operator, the genes can no longer be transcribed. Okay? So it means the presence of tryptophan, because tryptophan iko tayari, so hakuna haja ya kutengeneza ingine. But inafanya aje so that the bacteria cannot make tryptophan when it is not actually needed. That tryptophan stops the operon by binding to the repressor. Once it has bound to the repressor, it activates that repressor protein. The repressor protein is a product of the regulatory gene. So once tryptophan binds to the repressor protein then now goes and binds to the operator. The repressor operator formation is what now blocks the operon from being transcribed by the RNA polymerase. Once the RNA polymerase cannot transcribe the genes, then transcription has been stopped. Once there is no transcription, there can be no translation, because translation works on the product of transcription. So in that case, the cell stops making tryptophan just because there is already enough of the tryptophan within the environment of that particular cell. So in that way, the trip operon, which is a repressible operon, has helped to save wastage. There is no point for the cell making more tryptophan when actually there is already enough of it in the environment. So the presence of tryptophan acts to stop, to turn off this type of operon. Okay? These are diagrammatic representation. So when tryptophan is present, it will lead to the binding of the TRP repressor onto the operator. Once that has happened, then the RNA synthesis is blocked, the transcription process is blocked because the RNA polymerase cannot move. Okay? The repressor present on the operator inakinga barabara ya the RNA polymerase. So there can be no transcription. But in the absence of tryptophan, then the repressor dissociates from the operator. Dissociating ni kutoka, inawachana na operator. Repressor ikitoka kwa operator, then barabara ya RNA polymerase iko clear. Now the RNA polymerase can move and transcribe the structural genes. Okay. So the key points here, what we need to remember is that for a repressible operon, it is normally on, but it has to be turned off when there is enough of a particular molecule in the environment. So there is no point of creating more when actually you have enough. What helps to turn off this operon is a co-repressor. In this case, something like the amino acid tryptophan. The co-repressor inaenda bind kwa repressor. Then the repressor becomes active. Once the repressor is active, it now goes and binds to the promoter, to the operator. Okay? The active repressor binds to the operator. Once there is an active repressor on the operator, then the RNA polymerase yenye itakuwa imekuwa bound ama imeshika kwa promoter, the RNA polymerase that will have attached itself to the promoter will not be able to move, because remember, you come from the promoter to the operator before you get to the genes. So hata kama RNA polymerase iko kwa promoter, lazima whatever is on the operator itoke before the RNA polymerase can be allowed to move. But if there is a repressor that is activated on the operator, then the RNA polymerase on the promoter inakuwa na barabara ambayo imekingwa. Sasa haiwezi ikapita, ikaenda transcribe the genes. So the result of all this then is that genes for making tryptophan are turned off, are switched off when there is enough tryptophan in the environment. Okay? So just as a quick summary, when there is low tryptophan, the operon is turned on. The genes are active, so they can now make more tryptophan. When there is high tryptophan in the environment, tryptophan acts as a co-repressor by binding to the repressor. Once the tryptophan repressor complex is formed, it now goes and binds to the operator. By the tryptophan repressor complex attaching itself to the operator, it will block the RNA polymerase from transcribing the genes within the operon. The result of that is that now those genes are sort of turned off. They are switched off. Okay? So by this way then, the cell is able only to make tryptophan when it really needs to. Okay? So when there is already plenty of tryptophan, then the cell does not need to waste energy in making more of it. Sababu iko tayari available. So hakuna haja ya kutengeneza ikini. Okay? So that is how prokaryotes, bacteria, regulate their genes. How to turn on a particular gene, how to turn off They do so through operons. The operons come in two types, an inducible operon and a repressible operon. Each of these operons has a promoter, an operator, the structural genes, a regulatory gene. Okay, it has all those four sections. So thank you very much. Listen to this video, read the notes, and should you have a question, feel free to reach out to me. Asanteni and all the best. Thank you.